the outro. So Tony, you can share your slides again. And in the meantime, I will like to say that it's a real pleasure for us to introduce the first speaker, that is Dr. Tony Hunter, that is a pioneer in cancer biology. He discovered the tyrosine phosphorylation, understanding how a signal transduction is driven by tyrosine phosphorylation and how is it involved in cancer. So today he will be speaking on histidine phosphorylation, and again, we are truly honored to have him with us. So please, Tony, the stage is yours.
Thank you very much. It's a pleasure to do this. So, as you all know, protein phosphorylation is a major cell regulatory mechanism, and much of what we know about phosphorylation is through our studies of serine, threonine, and tyrosine phosphorylation, so-called classical phosphorylation, which generates heat-stable linkages and is strongly implicated in disease, including cancer. But you may not all know that 6 other amino acids can be phosphorylated. 3 basic amino acids, arginine, lysine, and histidine. The 2 acidic amino acids, aspartate and glutamate, and cysteine. And these non-canonical phosphorylations generate phosphate linkages that are heat and acid labile. And we know very little about their functions and whether they're involved in diseases such as cancer. So I'm going to talk to you about our work on histidine phosphorylation. Histidine phosphorylation was discovered over 60 years ago now by Paul Boyer's group in 1962, who demonstrated that there's a mitochondrial enzyme that contains phosphohistidine that is used as an enzyme intermediate. And you can see over the years there have been many advances in our understanding of histidine phosphorylation, including target proteins, other enzymes that use phosphohistidine as intermediates, discovery of histidine kinases and phosphatases, and some examples where histidine phosphorylation regulates protein activity. And most recently, efforts to define the global phosphohistidine proteome. So this was the first paper from Paul Boyer's group in Minnesota, describing identification of phosphohistidine as an intermediate in OXPHOS. And he went on a couple of years later to show that the phosphohistidine was an enzyme intermediate in succinate thiokinase, perhaps better known as succinyl-CoA synthase. So for those of you who don't remember the structure of histidine, it's somewhat similar to that of tyrosine, where phosphate is added to the hydroxyl group of the phenolic ring. Histidine has an imidazole ring with 2 nitrogens, and it's unique among phosphoamino acids in that it can be phosphorylated at either the N1 position or the N3 position, in both cases generating phosphoramidate linkages, phosphorus-nitrogen bonds, which are both acid and heat labile, making phosphohistidine challenging to study. So the question I want to pose is histidine phosphorylation used as a reversible protein regulatory mechanism in which a histidine residue in a protein is phosphorylated by a kinase, generating either the 1 or 3 phosphoisoform. And this is then reversed by a phosphohistidine phosphatase, converting the protein back to its basal state. Now, we know in bacteria that histidine phosphorylation is used for regulatory signaling, in which there's a large family of receptor-like proteins that have a histidine kinase that autophosphorylates on a histidine. That phosphate is transferred onto a second protein, a response regulator, onto an aspartate residue, which gives you the biological output. And this system is used by bacteria to sense many different types of external stimuli. And just in case you're skeptical about phosphohistidine, there are structures with phosphohistidine. This is the Ki-A sensor kinase of one of these 2 component systems, where you can see the 3 phosphohistidine residue is created as the intermediate. Now, in mammals, we've lost these two component systems. In fact, they're only present in very simple eukaryotes. But there are two proteins that have been reported as histidine kinases that themselves use a phosphohistidine intermediate, and at least 3 different phosphohistidine phosphatases. So the two kinases are better known as nucleoside diphosphate kinases, or NDPKs. They're totally unrelated to the serine, threonine, and tyrosine protein kinase families. They use a 1-phosphohistidine intermediate either to regenerate a nucleoside triphosphate or to phosphorylate proteins on histidine and perhaps other amino acids. And this is taken from a review I wrote a few years ago now, just showing you the histidine kinases that have been reported, some of their substrates, and the phosphatases that oppose the kinase with at least 3 distinct families. Now, there's one reasonably good example of how histidine phosphorylation can control protein function in this work from Ed Skolnick's group on this calcium-activated potassium channel, KCA3.1, in which, in this case, in a T cell, the ion channel is normally closed through chelation of a copper ion by the 4 cytoplasmic tails on histidine 358. In response to T cell activation, there's a burst of intracellular calcium which binds to the calmodulin associated with the tails of this tetrameric channel. And this results in partial opening and phosphorylation of this histidine by the NME2 or NDPK-B histidine kinase. This displaces the copper ion, allowing the channel to open. The process is reversed by one of the phosphohistidine phosphatases, PHPT1. So there are quite a few examples of where histidine phosphorylation may play a role in mammalian cells. We have these ion channels that can be regulated. There's involvement of histidine phosphorylation in G protein coupled receptor signaling. Histone H4 is phosphorylated, particularly on the N-terminal tail on histidine 18. As I show you, there's some evidence that histidine phosphorylation could be involved in centrosome and spindle pole function. So how can one study this chemically very unstable modification? Well, we thought by analogy with phosphotyrosine antibodies, we might be able to use the same approach and generate phosphohistidine antibodies. And we tried this over 30 years ago now and failed, largely, I think, because phosphohistidine-containing proteins are simply or the phosphate is simply too unstable to survive as an antigen. And so we realized what we needed were chemically stable analogs, but we had to wait until some chemists, particularly Tom Muir and Mike Webb's group, generated these analogs, the phosphotriazolyl alanines, in which the phosphate, either in the 1 position or the 3 position, is linked to a carbon atom in a 5-membered ring that sort of mimics the imidazole ring. And so we use these analogs incorporated into degenerate alanine and glycine peptides to immunize rabbits when coupled to KLH to generate sequence-independent antibodies. And this work was published 10 years ago now, so I'll just briefly show you what we got. We were able to generate both polyclonal and monoclonal antibodies, and here are some blotting experiments done with, in this case, pancreatic cancer cell lines, where we have 2 lanes for each cell line, 1 lane where the sample was not boiled and 1 where the sample was boiled to discharge the phosphohistidine. So it's very heat labile. You can see the major bands we actually detected were the NMEs and perhaps some other proteins whose signal goes away. Where we used our 3 phosphohistidine monoclonals on either an E. coli lysate or a 293 cell lysate, again with a heating control, you can see a large number of bands that disappear with heating a sample buffer, some of which we actually know what they are. So we've been able to use these antibodies for immunoblotting under special conditions, IF staining, immunohistochemistry, and for affinity enrichment of phosphohistidine proteins and peptides for mass spec analysis. We actually solved the structure of 5 of our antibodies bound to these phosphorotriazyl alanine peptides. And what this taught us was how antibodies can discriminate against the 1-phospho form of histidine and the 3-phospho form of histidine. And here's just one example with a sort of a higher resolution picture of how the phosphate on the 3 position of 3-PTZA is coordinated by glycine XX glycine loop, as is the case for some nucleotide binding proteins. We've actually used our structural information to try and engineer higher affinity derivatives of these antibodies. And here is one example of the SC44 3P-His antibody, where the original antibody shows a much higher affinity for 3-PTZA than for the authentic 3-phosphohistidine. It's about a 50 or 60-fold higher affinity. And through engineering, by making these 5 point mutations through phage display and random mutagenesis, we obtained an antibody, this 20N32F, which has about an equivalent affinity for 3-PHIS and 3-PTZA. The structure shows one of the reasons for this increased affinity, which is this phenylalanine mutation here, which forms a perpendicular pi-pi bond with the in this case, with the TZA ring here. And we've done some quantum mechanical modeling on why the antibody has these modified antibodies have higher affinity for the phosphohistidine. And we think it's because the phosphate on the phosphohistidine has a lower negative charge when it's bound. So here's an example of using our antibodies to stain cells, a particularly dramatic example where we've stained a mitotic HeLa cell showing very strong green staining with this 3-phosphohistidine antibody of the mid-body and the spindle poles. And we've also used these antibodies to affinity enrich. So we immobilized 6 of our antibodies and used them to pull out proteins from 293 T cells, where we had generated fully denatured lysates in 6 molar urea, diluted them down to put them over the antibody affinity column, and then elute at pH 11 to retain the phosphohistidine. And we got about 800 proteins that were enriched more than 2-fold over a control. Interestingly, about 400 of them had Go terms predicted to be involved in some form of RNA biology, which is of interest. And then about 100 had cell cycle Go terms, as our staining had suggested. And we found proteins that we expected, which we knew already contained phosphohistidine. Now, this doesn't map the phosphohistidine in these proteins, and we needed to do more conventional phosphoproteomic analysis, which is challenging because for enriching phosphorylation, threonine, and tyrosine peptides, one uses strongly acidic conditions, which can't be used for phosphohistidine. And so again, we resorted to our antiphosphohistidine antibodies to capture phosphohistidine-containing tryptic peptides from digests of total cell proteins, and then subject them to positive ion mode mass spec analysis, which again is an issue because the phosphate is discharged by bombardment in the instrument, and so one loses, in some cases, the information about the site of phosphorylation. In any case, Kevin Adam in the lab was able to do this using slightly modified conditions for mass spec. And we obtained around 70 potential sites of histidine phosphorylation. Here's the pile-up sequence logo. There's no consensus motif, so far at least, for histidine phosphorylation. And you can see enrichment of leucine residues and basic lysine and arginine residues. And we don't know whether this is because this is a preferred sequence for phosphorylation or whether instead it's because these are the phosphohistidine peptides that are chemically more stable. You can see there are a number of potentially interesting proteins that I'm showing here, including one that we knew we should get. This is phosphoglycerate mutase that uses phosphohistidine 11 as an enzyme intermediate. And here's the LCK kinase. We have sort of reanalyzed our data and found two of significant interest that we think we might follow up on. These are proteins involved in nucleic acid recognition. And one interesting possibility is then that phosphorylation of an RNA binding protein like HNRMPM might regulate RNA binding by serving as a negative displacement regulatory system. So how abundant is histidine phosphorylation in mammalian cells? In some other organisms, it's been estimated to be 5% of total phosphate. Others, like Claire Ayers at the University of Liverpool, have developed other methods for enriching phosphohistidine and reported a few years ago now identifying hundreds of phosphohistidine and arginine, etc. sites. Other groups have also been in this area, particularly Lemire's group, and they've identified many phosphohistidine sites in cells. And then Richard Muimo's group has used NMR analysis in a lysate of human bronchial epithelial cells, looking for a unique phosphohistidine peak in the NMR spectrum, 3-phosphohistidine, comparing it to the level of phosphoserine and threonine, and has estimated that phosphohistidine might be 20% of the total of phosphoserine and threonine in a cell, suggesting that the hidden phosphoproteome could be a significant fraction of all phosphate linked to protein. But I should point out that Simone Lemire's group published a paper now 3 years ago reporting that the only phosphohistidine sites they could detect in HeLa cells using a stringent method were just 4 phosphohistidine enzyme intermediates. So what are the open questions about histidine phosphorylation? So what type of functions might be regulated? I've always suggested the cell cycle, RNA processing and translation might be targets. How big is this phosphohistidine proteome? It could be hundreds of proteins. Are there other histidine kinases and phosphohistidine phosphatases we don't yet know about? Is there selective phosphorylation at the 1 or 3 position? It looks like most of them will be on the 3 position. Are all of the histidine phosphosites that one can identify functionally significant, or are some phosphohistidine sites adventitious, as has been suggested for some serine, threonine, and tyrosine sites? And how are phosphohistidine signals decoded? Perhaps the most important question, is there a phosphohistidine binding domain like an SH2 domain? And then what would histidine phosphorylation actually do to protein function? Well, we have a suggestion it might inhibit metal ion binding because many metal ions binding sites have a histidine. We think it might regulate ring domain E3 ligase activity because one of the coordinating zinc motifs contains a histidine in ring domains. We've been interested in the question of whether cancer might be driven by aberrant histidine phosphorylation. We've looked at several different types of cancer. I'll just tell you a couple of very brief stories. In liver cancer, we collaborated with Mike Hall's group in Basel, who developed a model of hepatocellular carcinoma where both PTEN and TSC1 are conditionally knocked out in the liver to activate mTOR signaling. This causes the development of multifocal tumors within about 3 to 4 months. And what he noted was that by doing proteomics was that the levels of one of the histidine phosphatases, he was focusing particularly on phosphatases and kinase, LHPP was extremely low in these tumors, and that led him to contact us to collaborate using our phosphohistidine antibodies. And you can see that when they analyze tumors compared to the normal liver, samples, they saw an increased level of 3-phosphohistidine and 1-phosphohistidine in NME1 and NME2. So it's about a 2 to 3-fold increase in both of these. Again, there was a boiling control in each case. They went on to show by re-expressing LHPP in the liver of these mice using an AAV vector, that this decreased the number and size of tumors quite significantly, suggesting that LHPP might be acting as a tumor suppressor. And importantly, they went on to show that in hepatocellular carcinoma patient tissue that there was high levels of 3-phosphohistidine, 1-phosphohistidine, and here you can see the very low levels of the LHPP phosphatase. So again, a 2 to 3-fold increase. So taken together, this indicates then that both the human and mouse system hepatocellular carcinomas have increased levels of phosphohistidine, and the LHPP phosphohistidine phosphatase levels are low, suggesting it might act as a tumor suppressor, raising the possibility that one might be able to target histidine phosphorylation pathways in this cancer. And in pediatric neuroblastoma, the most aggressive forms have amplification of the NME1 and NME2 genes on chromosome 17. And this we were able to show together with Pete Sage's group at UCSD have highly elevated levels of 3-phosphohistidine. And we've been able to identify some proteins by using antibodies, anti-PHIS antibodies, to pull down proteins and look and see which of these are associated with the NME1 kinase. And there are about 30 proteins of interest. And we're following up on these two proteins, the PHB proteins or prohibitins, which have been shown to have possible roles in invasion. And unfortunately, I don't have time to tell you more about that story, which is in preparation for publication. We've also found that LHPP levels are, instead of being lower, are higher in triple negative breast cancer, and the LHPP knockout reduces TNBC tumorigenesis in xenograft models. And in pancreatic cancer, we found that the levels of the enzyme intermediate of ATP citrate lyase, which generates cytoplasmic acetyl-CoA, are increased in the CD45 myeloid suppressor cells in pancreatic tumor stroma. So we have some evidence then that histidine phosphorylation might be elevated or altered in different types of cancer. There's been huge success in generating tyrosine kinase specific inhibitors, or TKIs, as a major new class of cancer drug, with, as of the beginning of the year, over 85 tyrosine kinase inhibitors approved for clinical use. And so what we hope or imagine in the future it might be possible to develop small molecule inhibitors that block histidine kinases in cancers such as liver cancer and neuroblastoma, where it might act as a driver mechanism. So to end up, then I'll just discuss what I think is going to be needed really to validate histidine phosphorylation as a reversible regulatory mechanism. We have significant lists of phosphohistidine sites, and some ideas about histidine phosphorylation might regulate protein function, such as inhibition of metal binding or regulation of E3 ligase activity. What we really need to do is to validate the sites of histidine phosphorylation that have been identified by mass spec. The lists that have been published by Claire Ayers and other groups, as well as our bioarchive paper, don't really overlap significantly, which is somewhat of a concern. And so one really needs to validate these sites by making knock-in mutations. Asparagine has been used as a non-phosphorylatable analog of phosphohistidine because it's isosteric, but it lacks the positive charge on histidine. There's really no good phosphohistidine mimetic mutation. What we and perhaps others are trying to do is generate non-cleavable phosphoromimetic analogs to incorporate p-his sites using expanded genetic code technology. Hopefully this would be a way to determine functional consequences of histidine phosphorylation. I think it's important to generate sequence-specific anti-phosphohistidine antibodies, so you can test for changes in phosphohistidine levels in a protein of interest. We have made antibodies against the phosphohistidine 118 site in NME1 and NME2, which work. But really one needs monoclonal antibodies. And we're hoping to use phage display technology, the sort we use to increase, get higher affinity antibodies to do this. Then ultimately one wants to assess the physiological effects of phosphohistidine site mutation using knock-in mutations on functions where the protein of interest might be involved. So with that, I'll thank the people who did the work I've described today. Steve Foos was the brave postdoc who undertook to make the phosphohistidine antibodies. Several of the studies that I've described were done by postdocs, more recent postdocs in the lab. We had help originally with the chemistry to make the TZ analogs from Jacques Marget's group. We've collaborated with Mike Hall on liver cancer, Ed Skolnick on the ion channel, Pete Sage with neuroblastoma, and the antibody structure determination and phage display were carried out in collaboration with these individuals. So with that, thank you very much, and I'm happy to answer questions.
So thanks a lot for the fantastic talk. And so we can open the discussion. Do the audience has any questions? Okay. Go, Julian. I'm not able to unmute you.
Yeah, no, it's all good. I can speak. Yeah, thanks for the talk, Tony. I thought that was super interesting work. I was wondering if you could weigh in on the recent study, the Nature Methods paper that was much more stringent and identified very few phosphohistidine sites. Do you think they're being too stringent? Do you think, yeah, essentially, how do you view that?
Yeah, I don't have any quarrels with the data they presented. I think they missed a lot of known phosphohistidine sites. So clearly there's some issue. Maybe they are being too stringent. They didn't find phosphoglycerate mutase, for instance, which we found fairly easily. So there are several other enzymes they should have found if they were doing a global survey. And we know from our, blotting studies and identification of other phosphohistidine proteins that there are other phosphohistidine sites. I mean, in their original paper, they suggested our antibodies and other people who made phosphohistidine antibodies are cross-reacting with other residues. And I think we've pretty clearly shown that it isn't the case. So I think that, we need to do more work. And as I said at the end, we definitely need to validate these sites by knocking them out and showing the signal goes away. I think that's really the only correct way to do it. So I'm still optimistic that that there are a significant number of phosphohistidine containing proteins where there's potential for regulation. But it's certainly an unanswered question, I would say.
No, absolutely. Thank you.
Okay, more questions? Okay, I may have one. So we are in the era of artificial intelligence. So do you think that in some way some machine learning algorithms trained on phosphotyrosine, phosphoserine, threonine could help in identifying some high probable sites that then could be used to generate some knockouts model or some knock-in? So to prioritize some list of, some mutants that you can further study.
So it's a good idea, and I'm sure artificial intelligence algorithms will help us figure this out. I think it will be interesting actually to put in all of the reported phosphohistidine sites, whether or not how authentic they are, into a search mode, a search algorithm, and see what it would come up with. I think we are entertaining the idea of using a degenerate histidine containing peptide library to see if we can use NME 1 and 2 to get a consensus sequence for phosphorylation, which will be a little different from looking at identified sites of PHIS, where the identity of the phosphorylated residue may not always be correct. So this will be a peptide library approach of the sort that Cantley's lab pioneered. And so that might be one way to see if there's any sequence context for histidine phosphorylation. Most of the sites that we've really looked at seem to be in unstructured regions, so it doesn't seem to be a structural basis, but there could be a sequence basis. I think,
So
yeah, it's something that would be helpful.
So in some case, like large language models could help. I don't know how, but in some way, like cracking.
Yeah, I think a lot of people are beginning to use them to try and unravel phosphorylation networks, and
yeah, definitely.
and then histidine is only one of the 6 non-canonical amino acids, so there's a lot of potential here. I mean, in bacteria, there's a very well-developed arginine kinase and phosphatase system that is involved in regulation of metabolism. So,
yeah,
I think there's a lot more to be uncovered here.
Yeah. So, Jesus, you can unmute yourself. Yeah.
Hi. Do you know, Tony, if there's any example, or would you imagine that it is actually possible that some viruses will also need this modification for other proteins?
That's a very good. No one's asked me that question before. I'm sure that viruses might have decided this would be a good regulatory system to target. I just don't have any evidence that that's the case, but it would probably be, one of the large DNA viruses which have plenty of coding capacity to evolve something that perturbs histidine phosphorylation, or maybe bacteria. In fact, pathogenic bacteria might also decide that this will be a good cellular function to target.
Nice.
Okay, if there aren't any further questions for Tony, we can move to our next speaker, which is Jesus.